We'll continue our discussion of phylogenies and go into a little bit more depth about types of phylogenies, how to read and interpret phylogenies, and then look at some of the terminology that's used as we base our classifications of organisms on these phylogenies. So phylogenetic trees, sometimes just called trees, are like mobiles. And I use this term just because I think it's a good visual, although maybe you're not super familiar with a mobile. I made one, I think, in like fourth grade as an art project, but I think more commonly you see them like hanging above babies' cribs, right? Where you've got a central attachment at the top and then a little two wires or something coming out and then a couple more things hanging down and then sometimes subsequent branches under that. So think of it like an upside down mobile where this would be the hanging part. And the reason I use this analogy is because mobiles often, I think almost always have like the ability to twist and move. The wind or just the air can, sometimes there's a little motor on the ones above babies' cribs, right? So you can twist them around the individual nodes, which we're going to do here with this phylogeny. So notice I've got a phylogeny species A, B, C, and D. If I twist it around the bottom branch here, I end up with just the reverse, where A is on the right instead of on the left, and then B, C, and D. But notice none of the relationships changed. So C and D are still most closely related to one another. Then their next closest common relative is B. And remember, C and D are equally closely related to B. And then they're all together, their closest relative is A, and they're all three equally closely related to A. And so no matter which branch I twist, so if I twist that branch around, I got B and C. And so this is why you can't just say, oh, look, A is closely related to D on this phylogeny, but it's distantly related on this phylogeny. That's not the case, right? So it's not where they are next to each other, it's how the branches connect together. And so all three of these trees are identical. They're equivalent, right? Just by having a wind blow the mobile around so one part's closer to another part didn't change any of the internal connections. And the same thing is true with phylogenies. So in which cladogram are A and B most closely related to each other? The answer is, well, they're all equally closely related to each other in all three of these phylogenies because they are identical, right? So you should be able to look at a simple phylogeny like this and determine whether it is identical to another one despite twisting the nodes around, as long as we didn't change any of the branch connections. Now, if the branch connections change, right, that's a different phylogeny, a different hypothesis of relationships. So if I took branch C here and disconnected it from where it connects in with the ancestor of D here, and instead drew a line so it connected in with B, and so then B and C shared a more recent common ancestor with each other, then that would be a different phylogeny and would be saying something different about the history of those species. Now, sometimes we are unable to determine all of the relationships with a given data set, and we'll talk next week about some of the methodologies we use to figure out phylogenies, but we're not going to get into a lot of depth. But if those methods we're using to figure out relationships we have some problems with them, then we may end up with a polytomy for some or all relationships. Okay, and there are two types of polytomies that you need to be aware of. A hard polytomy is where more than two branches come from a single node. So in this case, I've got 5 branches that all simultaneously diverge from that same ancestral population. Now, what this means is that this is an actual reflection of the real history of these 5 species, that all 5 of these species diverged at the same point from a common ancestral species and then evolved to become their own different, distinct lineage. Okay, hard polytomies are probably fairly rare. Most speciation processes divide populations into 2 groups, and then they'll change enough to be considered different, and then there'll be another subsequent divide for the 3rd group and 4th and 5th and so on. But it's not impossible and does happen when we would have more than 2 descendant populations that all diverge from a single ancestral population at the same time. And when that occurs, it's called a hard polytomy. More commonly, when you see a polytomy in a phylogeny, it's actually a soft polytomy, which means we're going to draw it as if they had all descended, but that's not because that really happened. It's because we maybe have conflicting phylogenies. So maybe our data equally support 2 different hypotheses, and so we don't know which one is right, and so we have to collapse some of these nodes. Okay, and that can happen in parts of the tree or in all of the tree. And so typically, if we have a polytomy, the next thing to do is say, well, let's gather more data. Let's add additional species to our phylogeny. Let's sequence more genes. Let's investigate the physical characteristics of those species and make a data set that will allow us to resolve this soft polytomy. So soft polytomies are saying, well, we're not certain, we don't know. But if we gather more data, we can potentially resolve a soft polytomy and find out which of the many possible phylogenies are the best supported by that additional data. Okay, so scientists are continually building data sets, gathering more data, and trying to resolve polytomies. Now, if we do it over and over and over again, and we continue to get a polytomy despite our data being gathered and more being added to the analysis, at some point, then we probably need to just say, well, it most likely is a hard polytomy. We can't resolve it because there really was a simultaneous divergence, and no matter how much data we gather, we're not going to be able to get any more resolution to the question because this is a reflection of reality. Okay, so just be aware of those two types. A polytomy is any part of a phylogeny where more than two branches come from one node, and that hard polytomies reflect the true history of the groups. Soft polytomies represent uncertainty, and we may need to add data to fully resolve it. There are also three types of diagrams. We'll really mostly only be working with 1 and 2, a cladogram and a phylogram. But I do also want you to know the definition of ultrametric. It comes and becomes important for molecular clocks. Okay, so a cladogram is a tree or a phylogeny that only shows relationships. The branches are just drawn at whatever length is convenient to make it regular. And so you'll almost always see all of the species on the same line. If it's drawn from bottom to top, you'll almost always just see them on a line at the top. If it's drawn from left to right, which some phylogenies are just for convenience, there's no difference between a phylogeny that's pointing up and one that's pointing to the right. But if it's a cladogram, you'll see all of the end species all kind of on the same line, and so the lines are just there for convenience. However, sometimes you'll see a phylogeny where the branches are different lengths. So notice here this branch leading to NAV18, I think these are viral samples, but that branch is longer than that branch, and those two don't have really any length at all other than just to say that they're separate, they're different from one another. So in a diagram with different branch lengths, we call it a phylogram, and it's a type of a phylogeny where the branch lengths are indicative of the amount of evolution that's occurred. And so because of the long branch lengths, we would expect these two viruses to be very different from virus V18 because there's lots of branch lengths, lots of changes that have occurred. And we would expect these two viruses to be really similar to one another. Okay, the branches that separate after a node, the vertical branches here in this diagram, they don't mean anything except to separate. So when we're talking about branch lengths, we're talking about length from the tips to the root. And we'll talk about roots in a little bit, right? So that's the branch length we're talking about. And so notice that these are slightly different. I think that one's a little bit shorter, but they both evolved quite a bit. Now notice this one, NAV1-2, has evolved very little since it diverged from its common ancestor. There's no length there at all. And NAV1 has evolved quite a bit since it broke off from that common ancestor. But they still have the same common ancestor that's separate from all these other common ancestors for the other organisms. Now, one other quick point about this phylogram. There is a polytomy on this phylogram. Previously, I showed you a polytomy when we have these triangular branches, and the shape of a tree doesn't matter. Whether it's drawn in a triangle or in squares like this, they mean the same things. But notice there is a polytomy here at the basal node where we've got this group and this group and this group, right? So those three groups all converge on this same vertical branch. That's the node right there. That's the common ancestor. And so in square phylogenies, rather than them all coming together, they just come together on a vertical branch, which are simply drawn to represent the common ancestor. So in triangular phylogenies, right where the branches come together, that's the node. In these square phylogenies, the connecting lines here, these vertical lines, that's what represents the node. And since all of these come into that same vertical line there, that means this is a polytomy where we are uncertain about the relationships between that group of species that group of species, and that one. We don't know how those three groups are related to one another. We know the relationships within those groups because those parts of the phylogeny are resolved. Okay, so that's a cladogram, only shows relationships. That's a phylogram, shows relationships and estimates amount of evolution along the branches. Now, an ultrametric tree is a type of a phylogram that is even more constrained. Okay, so sorry, branch lengths represent amount of evolution. Okay, an ultrametric tree has an additional constraint. And in ultrametric trees, the branch lengths, internal branch lengths, and the terminal ones still represent amounts of evolution, but we are constraining evolution to a constant rate. That means even after species have diverged, that their rate of change in evolution is going to be the same over here. And so to trace to any ancestor, if I'm going to the ancestor of A and B, they will have evolved the same amount by definition. If I'm going to go to the ancestor of A, B, and C, the amount of evolution that's occurred to get to B equals the amount of evolution to get to C. So notice those vertical lines now because we're up and down instead of left and right. But these lines here are the same length. That is an ultrametric tree. And ultrametric trees are a constrained version, usually occur only under specific conditions when we have neutral evolution and random mutations are accruing and building up in the populations at the same rate. So this is not going to apply to all data. But if it does, it's really kind of exciting because then we could use that data to estimate if we knew what the mutation rate was and we know how many mutations separate these species, we could use that as an estimate of how long ago these species diverged from one another. And that is a molecular clock estimation. So the branch lengths in an ultrametric tree still represent the amount of evolution that's occurred, but it's a constrained evolution that must proceed in the same route or at the same rate in all of the descendant species, right? So we've got cladograms, additive trees, and ultrametric trees, and we're just reinforcing that that left to right, if it's drawn if the tree is drawn from the bottom up, the left and right is not important. It's just whatever is necessary to create enough space for the diagram. So these represent the node, but the length of it is not important. It's the branch lengths from the tips to the root, the bottom. That's important. So now let's talk about roots. A root is simply the most ancestral node in a diagram, the most ancient ancestor that's represented by those species. So here I have a rooted tree. They've highlighted the root here. Usually it'll just be a single line drawn down there representing the root, but they've highlighted it here. So that is the root of this tree. This one I think is for primates, humans, chimpanzees, gorillas, orangutans, and baboons. Excuse me. So roots are important because they tell us the sequence of evolutionary events, which things happened first. So in this rooted tree, we know that the baboon split off from the other primates before the orangutan did. So baboon split off first, then the orangutan, then the gorilla, and humans and chimps are most closely related to one another. So we know the sequence of those events. Unrooted trees still show relationships, but they provide a little bit less information because they don't show us the sequence of events. Now, we can put roots on any branch. And so how do we determine if I've got a whole bunch of organisms, how do I determine where that root should go? Should it be here or here or here? It can go on any of those branches. So the way that we determine a root is by using an outgroup. And sometimes if we don't know what the single outgroup is, we might pick a variety of outgroups. So here I've got a baboon, an orangutan, gorilla, chimpanzee, and human. Maybe I choose a distantly related primate like a, I'm going to say sifaka, but that's the not technical, but it's not as common a name as a lemur, right? So if I chose a lemur, which is also a primate, but not closely related to these primates, then that would help me find a root. Now, if I was uncertain about the outgroup, I might pick 2 or 3 primates, and then even a species outside of primates. Maybe I pick something that I think is closely related to the primates, but not part of it, and that helps me establish the root for the remaining species. Okay, and so realize there's a little bit of extra work, or maybe even circular reasoning here, where we say, oh, to understand the relationships of this group, I need to include a close relative. But what if I don't know the relationship? So we do lots of work over and over again, and if we're getting consistent results, then we say, okay, I know that a close relative of group X is group Y, so I can include group Y whenever I'm doing a phylogeny of group A. I can include a member of group Y as my outgroup to help me establish and find the root for my study group. Okay, so that's how rooted versus unrooted trees work. We're pretty much only going to be looking at rooted trees in this class, but occasionally and in other classes you might see unrooted trees. So just be aware that roots imply the most ancient ancestor and then give us an order, sequence of events, a time, if you will, for the remaining part of the phylogeny. Okay, so there's some terminology, some vocabulary that you need to know, and most of you are familiar with this. If not, I apologize. Well, I shouldn't apologize. I didn't make up these words, but they're pretty awful. They're based in Greek root words, but they're long and sometimes similar to one another, and they are what they are. So you just kind of have to know them. The first three are not too bad. Mono, para, and poly all are fairly common prefixes, again, based from the Greek, but mono means singular, of course. Para means side by side, like parallel lines, and poly means many. And so that can help a little bit when we're trying to understand these. So I'm going to use this diagram here, but I'm going to kind of sketch on it here to illustrate these words. So let me just cut these out, and I'm going to go to a diagram where I can see it in better terms. So we're going to discuss these vocabulary words, and I'm going to illustrate them on a phylogeny. Okay, so these three words refer to classifications, groups of organisms that we have included in the same group and then given a name. Okay, so a monophyletic group is any group of organisms where we have included all of the descendants from a common ancestor. That's important. I'm going to say it again, and you can think about it. A group that includes all of the descendants from a common ancestor. So we have three different monophyletic groups. Well, four technically. Of course, any group of species, because all species share a common history. If I were to select all of the species, A, B, C, D, and E, and put them all in one group, it's always going to be monophyletic. So that's the easiest way to make a monophyletic group, is just put everything in and say, hey, they all share a common ancestor somewhere. Now notice monophyletic groups don't say anything about the smaller groups that might be contained within it. So there are three smaller monophyletic groups shown on this phylogeny. D and E together can be put into a group, and we could give them a name, and that would be a valid monophyletic group. I'm just going to put an M there for monophyletic, because we have included all of descendants from that common ancestor. Okay, we could put A, B, and C into a group, and that would also be a valid monophyletic group, because I've included all of the descendants from that common ancestor. And then the last possible monophyletic group is A and B together. That also would be monophyletic, because I've included all of the descendants from that common ancestor. So a monophyletic group really represents an ancestor, and then we're going to put all of its descendants into a common one. So for instance, this might be reptiles. Right, so maybe this is a pigeon, species E is a pigeon, and species D is a lizard. Now, why did I call those reptiles? Hopefully you know that, but we'll get into that here in a little bit. So that would be reptiles, or whatever other scientific name we wanted to give them. Right, and maybe this is mammals, so maybe we've got a kangaroo, a rat, and a rabbit here or something. And so this monophyletic group would be mammals. And then this one, the rat and the rabbit, I forget the name of that group. That includes reptiles, that includes the rodents and the lagomorphs, which is the group that rabbits belong to. But that is, I think they're closely related to one another. So anyway, that would be whatever name we give them. Okay, so all of those are monophyletic groups. Now, the only valid groups, the only valid named classifications are monophyletic, right? So for instance, if I took B and C and put them into a group together and gave that a name, that would actually be a paraphyletic group. I'll put PA. And it would be not a valid group. Now, in modern science, we don't do this because we use our phylogenies first, but many groups were done before we had good understanding of relationships. So many groups were organized and named, and then later, as we begin to understand relationships, we realize, oh, we've left out some of the members of this group. Okay, so notice that B and C, their common ancestor, is traced back to right there. That's where the B and C lines connect together. But we've left out one of the descendants of the common ancestor. So that is why it is a paraphyletic group, because we left out one branch. If I whacked off that branch, if they didn't exist, then it would be valid. But since they are around, we can't just ignore them. We would need to include them. And so we would fix the paraphyletic group, we'd make it monophyletic by including all of those in that group. Now, let's get into a little bit of history. This is the most famous example of a paraphyletic group. Originally, birds, we'll say group A here is the birds. Birds were placed in one class, and the extant reptiles, let's say alligators, I'll just abbreviate here, I can't write real well on the thing anyway. Alligators and lizards were placed in a group called reptiles. So these were placed together along with snakes and a few other things, and they were called reptiles. Later, as we gathered more data and did more analyses, and this was realized especially from as we got more fossils, even before we had good genetic data, but the genetic data has just confirmed it, that birds are more closely related to alligators than they are to the other living reptiles. Birds are actually part of the dinosaur group, right? So they're more closely related to the theropod dinosaurs than they are to other dinosaurs. So birds are dinosaurs, birds are reptiles, and so now they're included as part of that group. And so once we have enough data to support these analyses and we're getting confident in the relationships, we revise these older classifications that are not valid. So paraphyletic classifications are revised and are not valid. We cannot use them as a valid classification. Now let's handle this last one. Let me get rid of all these. All right, so I'm just going to put a little X here to remind us that paraphyletic are not valid. Polyphyletic, these do pop up also. Most often polyphyletic is because of convergence. Paraphyletic classifications like the bird ones often are the result of something called Synapomorphy, which we'll talk about here in a little bit. We'll come back to that. Sorry, I'm going to be slow here. Synapomorphy. Wow. My mouse penmanship is not real great. It's a long word too, right? Synapomorphy. Polyphyletic classifications are one of two things. Either people are like, for instance, it's a big group like beetles, and they're trying to figure out where to put certain species of beetles, and all of the ones that they just can't really figure out where they belong, they put into what's called kind of a trash bin classification group, where everyone knows it's not a good classification, but they just put them there for convenience sake until they can figure things out later. So that happens sometimes, but often polyphyletic groups are the result of convergence. Hopefully you're familiar with that term, but if not, we'll talk about it in a little bit here also. So convergence is the most common term, other than just, well, we don't know, so we'll put them in a kind of holding group. But convergence is the most common reason for polyphyletic. One example of an old polyphyletic classification that was due to convergence was the original classification for vultures. So we've got vultures here in the New World, condors, the black vultures that we get here commonly that you see flying out and sitting on the radio towers. But New World vultures, it turns out, once we gathered more data and evidence, and genetic data helped to firmly establish this. But the New World vultures are actually more closely related to the storks and a few other groups. So those New World vultures are more closely related to the storks. Oh, that's bad. I'm just going to get rid of that. And the Old World vultures that you see like in the African plains savannas, which look very similar, and they're adapted to similar niches, which is why superficially they look similar. But those Old World vultures are actually more closely related to raptors, the eagles, the owls, and the hawks. So originally, because of those similarities due to convergence, they were put in the same group and were called Vulturidae. Now this is polyphyletic because notice we've left out more than one branch. So any classification that you've got to start trimming off more than one branch, that's polyphyletic. That's a really bad classification. So these are not valid either. Okay, but sometimes historically they were put together because of these convergences. So for instance, if you looked at earthworms, and this is kind of silly because we realize we need a more in-depth study, but if you looked at earthworms and snakes, and there are some lizard species that are legless, and there's an amphibian group that is legless. If you looked at all those and said, oh, we're going to call them all, whatever group without legs, that would be invalid because you're taking a hodgepodge of groups, amphibians, some reptiles of different groups, and then annelids, which are not even vertebrates, right? And you're putting them in, trying to put them in the same group. Okay, so it's not common that we have really, really bad ones. Some of the very ancient classifications, the earliest ones before we even understood much about animals, were very, very poorly polyphyletic. But that's rare. Okay, but whatever it is, once we discover relationships and have a better understanding, we get rid of these paraphyletic and polyphyletic classifications and reorganize them so that all of our classifications, all of our name groups are monophyletic. Okay? All right, so now the next group of words we're going to talk about, these are also vocabulary words. They are also can be a little bit tricky, but they're necessary to help us understand and navigate the different types of characters, the evolution of traits on these phylogenies. Okay, so I've got a slide here. It's the same diagram, but I've got this slide with these vocabulary words here. So let's handle these, and I'm going to do it on the same tree here where I can sketch on it and talk as we go. Okay, so now we're going to handle these vocabulary words. So again, I apologize for the names. They're not mine. They're going to be similar to some other names too. So homoplasy looks like homology, but they're very different things. So don't confuse that with homology, which we've already talked about. Okay. So a synapomorphy is a characteristic that evolved in an ancestor, and all of the descendants still share that common character. So a good synapomorphy for mammals is production of milk to feed the young. All mammals have production of milk, and the ancestor obviously evolved that, and that has been inherited by all of the mammals. So whether you're a bat or a whale or a primate, you're going to feed your young milk to help them grow up. Okay, so that is an example of a synapomorphy. Okay, now a symplesiomorphy, excuse me, a symplesiomorphy is an ancestral characteristic that was lost in one or more of the descendants. Now remember I told you that birds were originally classified as reptiles because of symplesiomorphies. So the ancestral reptile was cold-blooded, walked around on 4 legs. It had scales and it had teeth, and there are a number of other things that we could add to that. The bird lineage lost many of those ancestral characteristics. Okay, whereas the alligator lineage, which is closely related to birds, didn't lose those characteristics. They still maintain many of those more ancestral ones. So that's an example of how a symplesiomorphy, right, can give you an example. But let's map these one by one onto the tree here. So synapomorphy. If A, B, and C represent mammals, I could put a line here and say this represents milk, production of milk for young. And that's a good symplesiomorphy because they all have it. Okay. What if I said hair? Right. That's another. Many groups have many synapomorphies that allow us to define. That's how those groups were defined originally. So those are all synapomorphies. But there actually are some symplesiomorphies for mammals also. One of the easiest ones. So let me do this. Let's say this over here represents reptiles. So I'm going to put abbreviations here. Bird, lizard. Let's make this a platypus, right? That's a mammal. So this group represents our mammals. This group represents our reptiles, birds and lizards and other reptiles. So platypus, let's say kangaroo, Ka for kangaroo. And let's just say an elephant. I could choose any mammals really to illustrate this point, but okay. Now, what if I asked you to tell me about the origin of the egg? Okay, meaning when I say egg, I don't mean the internal cells that are fertilized. What I mean is an external amniotic egg, let's say that. An egg that has a shell and a membrane inside the shell so that they can sit out in dry areas and not dry out and the embryo can develop. Okay, so lizards have eggs. We'll put a little check mark there. Birds, of course, have eggs. Platypus, yep, they lay eggs also. Kangaroo, they don't have these external eggs. Elephants don't have that external egg. Now, if these are the relationships, how do we explain the origin of the egg? Okay, now it turns out that there are 2 different possible hypotheses for this. One is that the egg evolved twice independently. Right? So it evolved once in the ancestor of all the reptiles, and that's why all the reptiles lay eggs. It evolved separately in the ancestor of the platypus. So that would be an example of convergence. Okay, but you need to recognize that there is another possible explanation. It's possible that the amniotic egg evolved much earlier, but then was lost. I'm going to draw a little line. The lines just simply represent an evolutionary change. We cross over the line as time passes on. We gain amniotic egg, and then this group loses that amniotic egg. Or maybe, if you want to be very technical, the amniotic egg loses the shell, and the amnion is now internal and becomes integrated into the placenta is attached to it, and you have it surrounding the embryo and all that other good developmental stuff. Okay, so this is different hypothesis. This is a sympplesiomorphy. And a sympplesiomorphy is where an ancestral state is then lost in one or more of the descendants. All right? So feathers are a great synapomorphy for birds. All birds have them, and they evolved in the common ancestor of the birds. That's if we're only looking at the living things. It turns out that actually synapomorphy, it's probably a better synapomorphy for the theropod dinosaurs, because many of those close relatives of the birds had feathers also. But anyway, if we're just looking at living species, feathers is a great synapomorphy, because all birds still have them. But what if I said the ability to fly? Well, the ability to fly did evolve in a common bird ancestor, but but it's been lost in many descendants, right? In penguins, in the dodo bird, in ostriches and their relatives, and there are many, many lineages that have lost the ability to fly. So flight is actually a sympplesiomorphy for the birds, not a synapomorphy. Okay, so be familiar with those examples that I've talked about. If I ever ask you whether something's a synapomorphy or sympplesiomorphy, I will either use a familiar example like we've talked about, or I'll give you a phylogeny with the characters on the phylogeny so you can look at them. Okay? Now, both sympplesiomorphy and convergence are forms of homoplasy. And homoplasy is simply a more complicated explanation. A synapomorphy is a very simple explanation. It happened once and then was never lost. Homoplasies are more complex. Either they happened twice independently, like convergence, or they happened once in the ancestor and then were lost in some of the descendants. And so because I have to mark two or more events onto a phylogeny, that is homoplasy, either as convergence or sympplesiomorphy. Okay, so be aware of those words. I understand they're somewhat similar. You just have to kind of know them, review them, and be able to recognize them in context given a phylogeny.